Our next presenter is Rama Khokha from the Department of Medical Biophysics from the University of Toronto. Rama is an internationally recognized leader in tissue homeostasis and stem cell niches. Her preclinical models and integrative molecular approaches continue to elucidate integral pathways in tissue development and remodeling. Her basic discoveries have revolutionized our understanding of cancer biology, allowed development of new strategies to block aggressive disease, and fueled breast cancer chemoprevention trials.
Good afternoon. I'm delighted to present at the RSC Café. A global rise in cancer incidence demands not only better treatments, but also new methods to preemptively intercept cancer. We study tissue homeostasis and apply understanding of breast biology to build new paths for breast cancer prevention. Apart from age, risk factors in breast cancer include family history, genetic predisposition, reproductive history, ionizing radiation, or increased exposure to ovarian hormones. For example, a well-known risk factor group carries mutations in BRCA1 or BRCA2 genes. However, treatment options for these patients are limited and include invasive procedures or few drugs that are not well tolerated. This motivates us to take on the challenge of chemoprevention, where the intent is to stop it from developing in risk groups. Conceptually, one can look at the breast structure as a dynamic ductal tree, which is bare or full of leaves. Just as these seasonal changes, appearance of mammary tree fluctuates regularly. We believe an ability to control this morphological turnover is at the heart of developing prevention strategies. After developing at puberty, breast undergoes cycles of cell expansion in response to ovarian hormones to prepare for a potential pregnancy and lactation. Remarkable changes from bare to full epithelial branches occur even within a single reproductive cycle, as you can see in blue for the mouse and in pink for the human. The cellular sites of interest are the leaf-like alveoli called TDLUs. These are the functional units for lactation and also the site where most breast cancers arise. Mammary ducts have two layers, each containing stem or progenitor cell subpopulations. These cells drive morphological changes and are also suspected culprits in breast cancer initiation. That is why we learned to purify and started to study such mammary cell populations. As a result, we found that TDLU-like structures cyclically develop and regress in response to the ovarian hormone called progesterone. We then went on to resolve how progesterone regulates stem cell dynamics by downstream paracrine signals and understood how these are precisely communicated across cell subpopulations. Our work has helped explain how cycles of stem cell activity can underlie breast cancer risk linked to ovarian hormone exposure. We have confirmed that progesterone-induced factors are causal in breast cancer, and including models representing high-risk patient groups. Others have shown that progesterone-related factors are higher in high-risk patients. Together, this has laid the foundation for two chemoprevention trials, which are currently evaluating anti-progestin and anti-rank ligand efficacy in BRCA1 mutation carriers. More recently, we have been able to gain molecular insights into these disease-relevant cell populations. In collaboration with our clinical colleagues, we have access to diverse primary breast specimens and have generated molecular maps on refined cell populations. This has produced a discovery resource for breast biology and breast cancer communities. We have uncovered unique lineage features, molecular signatures for cells of interest, and key pathways that are potential drug targets. To assess the therapeutic value of these pathways, we match our cell-specific targets with known or novel drugs and measure their ability to block unwarranted growth in morphology. Our systematic preclinical workflow provides us with go-no-go decisions to proceed to the next stage efficiently. and is ultimately allowing us to vet candidate preventive agents. While working with primary breast tissue, we have really grown to appreciate patient heterogeneity. Just as every tree is different, every woman is unique. Yet the framework of a mammary ductal tree follows a basic principle. In order to find the most widely applicable preventive strategies, it is important to understand what's common and what's different between patients. We now apply deep molecular profiling on a per-patient basis. We remain focused on cells purified from normal tissue and those from high-risk women. Technology advances even enable profiling at a single cell level. For instance, we can now profile all breast cells, including epithelial cells that I talked about, but also all the other cell types found in the breast, such as fat and immune cells. This has recently revealed that cellular composition of tissue from high-risk women differs, especially with respect to stem and progenitor cells. This knowledge will help us understand patient-specific variations, which is essential for future development of personalized chemoprevention. All of this work has been driven by different members of our fantastic team. I've been fortunate to have worked with so many academic trainees, outstanding students, and exceptional postdocs. On top of that, our collaborators have been absolutely tremendous. I would like to thank all of them and you for your attention. I also am very grateful to the funding agencies who have consistently funded our work for so many years. Thanks again.